I'm Irfan Ahmed. I'm the Executive Director of the Center for Nanotechnology and also the Executive Committee member for the Midwest Cancer Nanotechnology Training Center and iDrug. So I'm going to be introducing our two speakers today. It's 1 hour, so it's 30 minutes each, right? I mean, to do about 20-25 minutes.
I did
I
did a 15-minute, but wait, I can make it.
Yeah,
longer.
so 15 is fine.
Yeah.
So we'll leave another 10 minutes for question answer and then move to Eric. So Brian Wong is a PhD student in material science and engineering, being co-advised by
Professor Liu, Professor Helfrich.
And Professor Bill Helfrich,
Yes.
right? Yeah. So we'll let you introduce your topic and go from there.
Thank you.
Thank you.
All right. Good afternoon, as of 30 seconds ago. So my talk today is DNA-mediated synthesis of quote nanoparticles. We call them quote nanoflowers for diagnostic and therapeutic applications. So I guess we should maximize this. There we go. Okay, so whenever we think of DNA, I think it's still, outside of the research community, most people still think of DNA as a genetic material. In this particular talk, we're thinking more of it as essentially just a polymer. A polymer has some very, very special functionalities, very powerful functionalities, and actually very versatile. And so here what we see is the DNA double helix and kind of some of the highlighted parts of it. So you have the base, you have the backbone, you have the 5' end, you have the 3' end. And these are all things you can modify, change in many, many different ways during synthesis, post-synthesis, and to add a lot of very interesting functionality to it. So if we just look at it instead of a genetic material and look at it as a nanomaterial or nano object, what we see is that essentially in a double helix, essentially what we see is the width of it is about 2 nanometers. The length of it can be up to 50 nanometers in a rigid, so you can think of it as a rod up to 50 nanometers. Obviously, it can go much bigger than that, but after that, you're talking about multiple rods, you're talking about a softer, more flexible material. The structure is very, very well understood. We know exactly where every base is. We know the major helix, we know the major groove and the minor groove. We know a lot about this material. So, and then finally, one of the most important things is that actually it's very chemically stable. It can exist inside the body for a long time as a double helix, or as a hybridized form, not as a single-stranded form. And it's very biocompatible. So based on these kind of last three things, we can think of it as a very versatile and very interesting material to do a lot of projects with. So these are kind of things that people have done with DNA. It started out, it kind of goes from this to this side and then coming down in terms of chronological order. So people, Chad Merkin and a couple others, first started looking at DNA as kind of what's called smart glue. Essentially, it's just a really, really easy and really powerful way to bring 2 or more different nanomaterials together. Essentially, if you put a strand on one nano object, and a B strand on another piece of object, and you mix them in solution, and A and B happen to be hybridized, so they have the same sequence or opposite sequences. You can bring them together in solution in a very, very predictable and very controllable way. You can control the distances, you can control the number of functionalization on a nanoparticle, and therefore control the directions of how you attach the secondary object. And so a lot of work has been done with this, and our lab in particular turned this into many different types of sensors. So if you attach these things to gold nanoparticles, you can actually see the change, you can control the change from red to blue transition of aggregated and free nanoparticles in a solution. And we can break that, we can reversibly switch it based on the target. So in adenosine, cocaine, lead, uranium, mercury, we can turn this little vial of red or blue solution into a sensor, essentially, using the functionality that's built into the DNA as a linker. So that's kind of some of what my group does, a lot of people in our group does. So, but kind of moving away from that, we actually have something called structural DNA nanotechnology. And that's kind of where I spent actually most of my PhD, is in this kind of square, rounded oval thing. And this is something that we can spend a lot more time talking about if you're interested in it. So essentially the idea is, instead of hybridizing just to one strand of DNA, we can hybridize to many strands of DNA. And kind of hybridizing not only in a linear manner, we can hybridize it in a very, very well-controlled, well-thought-out, well-designed structure that it can be in 1D, 2D, or 3D. And so a lot of really cool structures have formed using these holiday junctions, using triple helixes, using triple crossovers, all these motifs that are essentially Lego blocks. And you can build almost anything out of these DNA. You can build cages, you can build cubes, you can build tetrahedrons, you can build buckyballs, you can build all sorts of really, really complicated things with these DNA structures. And one of the most recent and most kind of hot topic in DNA nanostructures is what we call DNA origami. Some of you probably heard of it. You take a very long strand of DNA and through some very, very simple programming, some very simple designs, you can fold this into a duck. You can fold these, people have folded it into a dolphin that has a little tail that moves. And these are all things that are made out of DNA. And you can kind of start seeing how powerful this structure is because every sequence on this DNA origami is unique. And therefore, you can address this piece of DNA uniquely in this tile. So you can put things arbitrarily and programmably on this tile in respect to anything else. So really a lot of cool things have been going on with this. What you see here is the 3-arm motif and 4-arm motif. So with the 3-arm motif, you can think that you can form a hexagonal 2D, or if you design it the way you want to, you can actually make it fold up and it will form a tetrahedron. With the 4-arm, you can form the square grid. And if you design it into a 3D, you can actually form the cube with these 4-arm structures. So moving on, another part of our group, it does a lot of this functional DNA, and something that I'll touch on a little bit later in the talk. So essentially, there's two types of functional DNA. There's a DNA zyme, and there is a aptamers. In this talk, we'll kind of focus on the aptamers part of the talk. Aptamers part of the functional DNA. So what it is essentially aptamers are the DNA equivalent of antibodies. Essentially, we select them in vitro through a kind of a combinatorial selection process to find certain sequences of DNA that somehow in 3Ds, in solution in 3D, they fold up some sort of conformation that will recognize a specific binding pocket, either in protein, in small molecules, even there's two ways to bind. It can either bind in the DNA or the DNA itself can bind to the protein. For bigger particles, the DNA will bind to the protein. And so this is a very, there's a lot of advantages in using DNA versus a protein antibody. It's much more stable, it's much easier to make, it's much cheaper to make, and it doesn't require, as far as we can tell, it doesn't have the same immunogenic response as a protein that's sometimes grown in rabbit or sheep or goat or whatnot. So these are completely, this is just a chemistry project. There's no other biological involved. So we'll touch on these a little bit more later in the talk. And then people have done some very, very creative things with DNA robots, DNA walkers, DNA. You can have a structure where in a linear, it's kind of almost a robotic assembly line where you have A, B, C, 3 different strands of DNA. You put the precursor to a chemical A, B, and C, and it can selectively, you can make it so that this robot will sequentially put A to B, then put A, B to C. And so it really offers a lot of really interesting applications that you really can't do with anything else. This level of control is not available with that many other materials. So if you have any more questions on any part of this, I'm more than happy to talk more about it. But the outline for today essentially is just, we're going to talk about these nanoflowers that I'm working on in our lab. Essentially, we'll give kind of a background on how we came about these nanoflowers, how do we do, just talk about the interaction between DNA and gold nanoparticles, talk about why these nanoflowers form, how these nanoflowers form. We'll do a quick slide on just how do we find aptamers, what are aptamers, and then kind of combine it all together to do into a system that we can talk about, we can use for hopefully some diagnostics and therapeutics applications. So, do you have any questions? I have to stretch this from like a 12-minute talk to a 30-minute talk, so questions?
You don't have to stretch it. You can.
Okay, so what we know is the DNA bases. There's 4 DNA bases, A, T, C, and G, and they're all chemically different. So this this group asked a very interesting question that a lot of people kind of knew already, but nobody really did anything about in 2002, which is, do these base pairs have any interactions with gold nanoparticles? Before this point, everybody would put a thiol onto the end of DNA and put them on the DNA, put them on a nanoparticle, and say, hey, I functionalized a nanoparticle. But what about the base? What about the rest of the DNA strand? It's much bigger. And although it probably doesn't interact with the gold surface as much as the thiol, it should definitely interact with the surface. And that's what they found. So this is kind of the hypothesis that if you have strong acting bases between the DNA and the nanoparticle, you have something like this. And this is actually less protective than what you can do with this. So if you have a less interaction between your DNA sequences and your gold, you should have something like this. And with this, you can actually, what's powerful about this part is that you can pack more DNA into a nanoparticle because you have, your DNA is not taking up gold surface. It's not laying down, taking up the gold surface. So what they found, this is kind of counterintuitive because they're looking at stability. So essentially what they see is that poly T base will interact least with a gold nanoparticle. So in this case, poly T actually gave it the most stable nanoparticle because the most DNA can get onto the surface of the gold nanoparticle. So based on this, we can see that, so they do AT and C because you can't really do poly G. Poly G gives you weird structures that is no longer linear. It forms G quadruplexes. So it's very difficult to synthesize poly Gs, and then you also don't really want to use poly Gs to stabilize nanoparticles. So they did AT and C, and essentially what they found was that T interacted much less with the gold nanoparticle. And A and C kind of did the same thing. Okay, so what does that mean for us? So these are kind of, I can jump again a little bit, but these are what we call the DNA nanoflowers. Essentially what we see is that if we take a poly A and poly C sequence of DNA, incubated with gold nanoparticles without thiols, they will adsorb onto the surface of the nanoparticle. And what we can do then is reduce additional gold onto the nanoparticle. And what that will do is grow the nanoparticle. If you have no DNA, you expect it to grow uniformly, or as uniformly as your control is. But that's not the case with A and C. So what we see with A and C is that when we grow it, it no longer is red like a spherical particle. It grows into these nanoflowers, what we call nanoflowers. So that was very interesting. And then when we did the poly T, it actually did grow into a spherical nanoparticle. So there's some different, we can definitely see based on this that there's some sequence dependence adsorption onto the gold nanoparticle that has now directed the shape of the gold nanoflower, the growth of the nanoflowers. We've actually taken this work to several different other nanoparticles. So in this case, we're using spherical seeds. We've also done using the nanorods as well as nanoprisms. And you actually get some very interesting sequence-dependent shape control using this technique. So instead of using poly A, poly C, poly T, you can actually start mixing. What if you do ATC? What if you do AAAC? What if you do AAAT? Something like that. And through just kind of a combinatorial library, we've found some pretty interesting shapes that this has also grown into. Lucas.
So will the body respond to the different shape controls, or is it much more responsive to general size control?
I think in this case, we don't know yet because this has not been in the body. We're trying to get it to that point. But it's so unknown. So what we've seen with the early cell studies is that cells seem to uptake these better, actually, than spherical nanoparticles, especially compared to DNA functionalized nanoparticles. So that's something that we definitely want to figure out. But so we can take a look and see that AAC nanoflowers, T and no DNA, you get spherical nanoparticles. You can grow. You can stop the reaction at several points, and then you can get TEMs and see kind of the growth sequence. So what you have now is we have our idea is that you've kind of swallowed up all the DNA, right? If all your DNA is lying flat on the gold surface and you grow the gold, you're going to swallow up all the DNA. Your DNA should no longer be available to the environment. So that's one thing we need to either confirm or deny. And what we did was essentially a very simple experiment. We take a smaller nanoparticle, put the complementary DNA. So if you have poly A, you put poly T. If you have poly C, you put poly G, something like that. And you incubate them together. So what you see is that when you have the poly A nanoflowers, your DNA is still available on the surface. And you can do the assembly, and you can, based on these images, you can see that after you form the nanoflowers, your DNA is still available to the environment to use. And so this is very important for moving forward. And then just using these nanoflowers, we incubate with cells, and we can see that they do uptake to the cells pretty nicely. I think these were incubated for 24 hours, and most of the cells were still viable. At that point, we hadn't done any specific toxicity studies, but everybody still looked alive, which is good. So a couple of things. Advantage of the system is that we do have a one-step. We can synthesize the nanoparticle and functionalize it all in the same step. It's very simple. We can get, so these are dark field scattering. So this is just an optical dark field scattering image. So we can get a very bright signal even from very few nanoparticles. And we seem from what we're just looking at now, it does seem to uptake better than spherical nanoparticles. And they're non-toxic because they're gold in DNA. So a couple of challenges we have is to look at directed uptake. We have to look at controlling the size of the nanoflowers, and then versus how that affects stability and uptake. And then seeing if we can tune the optical properties of these nanoparticles. So aptamers, this is how we get them. Essentially is a combinatorial selection. So you start out with 10 to the 14, 10 to the 15 different sequences of DNA. You just throw it at a target, see what sticks. You take the stuff that sticks, PCR it, randomize it, throw it at it again, and just repeat that about 30 times. And every once in a while, you get something that works. And then you can do negative selection. You can do a lot of other things to improve the binding and improve your chances. So aptamers just binds to a specific target. So in this talk, in this work, we're using AS1411, which is a aptamer binding to nucleolin, which is just a protein that's expressed on a couple of different cancer cell lines, including the MCF7 human breast cancer cells. So this is the design. So with the nanoflowers, so one of the problems is that we have to think about is that as you grow the nanoflowers, it does take up more and more of DNA because your DNA is on the surface. So as you grow the gold, your DNA is going to be less and less available. You can't grow it indefinitely because at some point you will have no DNA on the surface, and therefore it wouldn't work anymore. So the design is pretty simple. You have essentially what we call the gold surface binding the segment, which is A or C, poly A or poly C. I use the poly T in the middle because we know that it kind of desorbs from the surface, so you can use it as a spacer kind of. And then you have the aptamer sequence. So the hope is that if we can present these aptamer sequences outside of the nanoflowers and do some cell imaging, cancer cell imaging. So we can do. So this was something that there's two ways to control the size of nanoflowers. One way is to take one starting seed and just keep growing. Another way is just to use different seeds. You can start with a bigger seed and you don't have to grow as much. So that actually kind of gets us into a dilemma that I've talked about a little bit. But before I thought about this, I just used a 13 nanometer seed and just grow it bigger and bigger and bigger. And you can see a fairly well-controlled size trends, and you can actually tune the optical absorption of the gold nanoflowers. But this is the dilemma that I'm kind of dealing with right now, is that as your gold nanoparticles are bigger and bigger, it takes more. Your DNA does a less good job of stabilizing it, because your nanoparticle is getting bigger and your DNA is staying the same length, which is significantly smaller than the nanoparticle. So there is a couple of things that we're looking at to be able to stabilize these larger nanoparticles. And the reason we want to do that is because we found that at a particular size, we can actually get it to absorb NIR, which is something that we are interested in pursuing. So the work in progress now is there's a couple of things. We can increase the DNA density. There are several ways to do it that I can talk more about if you're interested. we can look at the ionic strength to increase the DNA packing, and then maybe even a post-synthesis stabilization, putting PEG on it afterwards, or putting other stabilizing molecules on the gold nanoparticle after we finish synthesize it. So the work I'm doing, so while that is going on, concurrently we're starting with the 13 nanometer seed and being able to show the aptamer uptake or aptamer selectivity. We do the, we just essentially, so this is the 13 nanometer seed growing to about 20 nanometer nanoflower, and then with and without the aptamer, so the control with a randomized DNA, and then with the aptamer DNA, we can see that there is we can see some selective uptake by the MCF7 cells. So the kind of where I'm going from here for next year is the most important, I think, is this actually second point, is to be able to stabilize the larger nanoflowers. That's something that I'm struggling with a little bit right now, but it is very important because that is kind of where the application of these will really come in, is that we can get them to absorb into the NIR, and then kind of to round out the experiment, we do have to very critically compare it to nanoflower or to nanoparticles, because that is definitely the accepted. That's what most people would use, and that's what Professor Merkin actually has a company on delivering these DNA functionalized nanoparticles for a lot of different applications. And then finally, if we can use it to, because we do have a functionalized nanoflower, we can use it to decorate it with other nanomaterials or cargoes. So with that, thank everyone in my lab. It's a big lab. Dr. Zedong now is, he's left us, but he started the nanoflower project. Hong, Li Hui, Kevin, Professor JJ's lab, Lee helping him with some of the cells, MRL staff, and thank you everybody for paying attention.
Thank you. Any questions, comments? Yes.
I had a quick question. The picture that you showed with the cells that were, or the nanoflowers that were binding to cell structure, you know, the cancerous cell and the healthy cell.
Yeah.
How does that bind happen? Is there any locomotion involved with the nanoflowers, or is it just?
It seems to uptake it. It does. It's not. So it doesn't seem to be binding to the outside. Mm-hmm. So if you see it, there is most of the nanoparticles are inside the cells.
Oh, okay.
Yeah.
Yes.
Do you think the shape of this nanostructure has any effect on the cell?
We hope so. I mean, we won't really know until we do some very, very systematic studies between the nanoflowers and the spherical nanoparticles, and hopefully we'll get to that point soon.
Do you think this unique structure will enhance uptake or decrease that?
Again, we're from what we know, just based on, because we haven't done the experiments, we're only comparing to literature values. These do uptake at the same concentration, or even 10 times less concentration than what the literature uses for nanoparticles. In particular, Professor Merkin's papers, that these we get better images at lower concentrations than they get at higher concentrations. But their nanoparticles are very, very, very stable, and they can use very, very high concentrations. So that's something that we're looking at.
Good. Anyone else? Okay, then. Thank you. Brian.